And we are online. Okay, so everybody, thank you. Welcome to this new installment of the International Legumes Society webinars. Today I'm happy to be here with Dr. Kristian Holst Laursen, which is associate professor at the University of Copenhagen, and it's leading of the research group on plant nutrients and food quality. And in fact, plant nutrients and food quality is the topic for today's webinar. He's also involved in the aptly named Peace and Love project, which I really like, and which is going on right now. And without further ado, I will leave the microphone to Kristian.
Thank you so much for the introduction, Nelson, and thank you to the Society for giving me the opportunity to present our work here today. So I want to start by acknowledging a couple of my colleagues who contributed to this presentation, to the slides. So that's Asger. I see he's also here today, Asger Sten Eskildsen. He's a scientific assistant. And then Jens Axel Knudsen, who's a PhD student actually on the Peace and Love project. So the topic is plant nutrients and food quality, and we've been working on this for quite a few years at the University of Copenhagen. However, not so many years in legumes. This is fairly new, but I'm going to tell you a bit about what we're currently conducting of studies regarding this. So first, a bit about our institution. So I'm from the University of Copenhagen, the largest university in Denmark, where we have around 36,000 students, 10,000 employees. It's a very international, you could say, university. So all our master educations are in English. So if you have any students or early career scientists interested in visiting us, then please do not hesitate. We're always happy to have exchange students and exchange of researchers as well. So the university is divided into 6 different faculties, and I'm located at the Faculty of Science. And the faculties are divided into a number of departments, and one of them is the Department of Plant and Environmental Sciences. So we are one of the largest departments with approximately 500 staff members. And we have 3 main areas that we're working on: environmental science, biotechnology, and then agricultural science. And it's mostly within agricultural science that I work and will be presenting today. So this is our campus at the Faculty of Science. It's in the center of Copenhagen, or at least most of it. So I'm located in this building here called the Copenhagen Plant Science Center. I'm assuming that you can see my pointer, right? Yes, you can. That's good. So I'm going to use that during the presentation.
Yes, we can.
Oh, that's great. So this is what we call Frederiksberg campus in the middle of Copenhagen. And as I said, I'm located in this new building called Copenhagen Plant Science Center, or it's actually also called the plant cell. You might understand why if you see it from above here. But to our campus and in particular our department, we do actually also have facilities outside the Copenhagen area, 20 kilometers outside of Copenhagen, where we are able to do our field trial studies. So we have experimental farms outside Copenhagen where we have approximately 200 hectares, where I'm going to present some of our field trial studies from a bit later on today. So this is also a part of our campus. So our department is divided into a number of sections, and within each section we have some research groups, and I'm leading one on plant nutrients and food quality. It's a fairly newly established group. It was established in 2019, and we are around 10 staff members plus a few students. And our aim is to bridge plant and food science. We are very interested in, basic plant nutrition. So what's the biochemical functions of nutrients? in plants. I've been working on this ever since I studied back in 1999. So I've been working on basic plant nutrition in many years. We are studying nutrient interactions, but also how various production systems, soil types, soil fertilities, fertilization strategies, etc., affect the plant nutritional status. And then we try to bridge this to the quality of the raw materials that we harvest, in particular raw materials being used for plant-based foods. And here we have a slightly different focus. So here it's the nutrient content, but also nutrient bioavailability we're looking into. We are quite often interested in the protein content, but also the protein quality. So for example, the amino acid composition. But in order to ensure that plant-based foods are safe, we also focus on heavy metals, for example, anti-nutrients, and we have even done study on food authentication. And quite often, the way we bridge plant and food science is by analytical chemistry, because we like to develop analytical methods that can be used within these various topics. So this is our analytical toolbox, you could say. In our group, most of our work is focusing on elements, the essential plant nutrients, but also heavy metals. So the periodic table is really where we start most of our work. Then we of course also study the plant available forms of various nutrients. So it's not only total sulfur, it's also sulfate. And if we talk about nitrogen, then it's nitrate and ammonium most often. And then we zoom in on elements as well and do stable isotope ratio analysis. And we use this, for example, to quantify biological nitrogen fixation, as I'm going to talk about a bit later today. So when we do stable isotope ratio analysis, it's mostly on nitrogen, carbon. We have also done quite a lot on oxygen. We have done studies on compound-specific isotope ratio analysis for tracing nutrients in the soil plant system, both of nitrate, sulfate, and phosphate. And then finally, we also do amino acid analysis and analysis of selected anti-nutrients. This week, for example, we are analyzing lysine and conlysine in faba beans and phytate in faba beans, peas, and oats. So yeah, and all the abbreviations here, they are basically the analytical techniques, instruments that we're using for our studies. So our mission in my group is to provide new knowledge and recommendations regarding sustainable and resource-efficient plant production. We want to ensure that it delivers safe, authentic, and nutritious foods. And we are not only working on legumes, but it is something that we are increasingly focusing on and have been focusing on during the past 5 years. So what is a sustainable crop, and what are we looking for in the transition towards more plant-based diets? Well, if the sky is the limit, then we would of course like crops with a low carbon footprint, and legumes are quite often stated to have a lower carbon footprint because they don't need the addition of synthetically fixed nitrogen. So this is quite often why these have a lower carbon footprint compared to, for example, cereals. We would also like our crops to have an improved utilization of the basic growth parameters, light, carbon dioxide, water, and nutrients. But simultaneously, we want to reduce our use of non-renewable sources. So in organic production, for example, we rely much more on organic fertilizers. And we have also done studies on, you could say, circularity, where we try to use wastes and industrial side streams for plant production, actually also in hydroponics. We want plants that are able to grow in a changing climate. This year we have had both a period of drought in Denmark and a period of too much water afterwards, actually. So we want plants that are resilient regarding these stresses. So we need resistance regarding diseases, pathogens, weeds, etc. And we are increasingly beginning to talk about stable yields instead of just higher yields. So it's quite important that the farmer knows that he's going to harvest something after each growth system, instead of just a growth year, instead of just focusing on getting as high harvest yields as possible. And here we are slowly beginning to bridge to the quality aspects, because we want to grow crops for food instead of feed, and there are some requirements regarding the quality. So you could say previously we would have said that now we have to produce more with less resources, but now we actually have to produce the right crops and the right raw materials with less resources. So from my point of view, this is the challenge today. So which crops are then suitable for this purpose? Well, it's interesting to consider that there probably exist several hundred thousands of plant species, probably more than 300,000. So we consider around 200 to be agriculturally relevant, but only about 10 plant species are being used for producing 95% of the world's food and feed. And wheat, rice, and maize are really dominating globally. It's slightly different in Denmark. Here we are growing barley, wheat, maize, grass, and rapeseed as the main crops. And 80% of our agricultural area in Denmark is actually used for feed. So there is not a lot of focus on crops for plant-based diets, and definitely not on protein crops. So Asger, who's also attending this webinar, he did this as one of his student projects, this graph on the production area of various crops in Denmark in a historical perspective, going all the way back to 1980 and then up to, I think it was 2021. And here we included wheat, barley, oat, pulses to maturity, so that's peas and fava beans, for example, and then peas to maturity and peas for consumption. And you should notice that the y-axis is logarithmic. So the largest area in Denmark has been and is still for cereals, so wheat and barley. And remember that in Denmark we have a total of 2.6 million hectares of agricultural land. So quite a large fraction of this is actually for cereal production. When it then comes to legumes, pulses to maturity, and so on, it has varied quite a lot during the past decade. So when I was a kid back in the 80s, there were peas everywhere. So there were subsidies for the pea producing farmers. Quite a lot of it was used for feed actually, and then it slowly dropped, going down to around 40-50,000 hectares in total in Denmark. And this drop was also due to severe incidences of soil-borne diseases. And of course, some farmers can still remember this, and this may be a reason why we don't have so many legumes in Danish agriculture today. So in total, we have around 45,000 hectares of legumes in Danish agriculture today, which is not much out of 2.6 million hectares, but it's steadily increasing. However, the majority of what we grow in Denmark today is still for feed, but we are doing our best to change this. And this here, I'm sorry it's in Danish, but it was in the Danish news. This is actually telling that there are new subsidies now for farmers who are growing protein crops and crops for consumption. So they can actually get what corresponds to, what is it, around 90 euros or something like this per hectare if they are growing crops for human consumption. So hopefully this is going to change this in the future. So what are the challenges in legume production? You are probably aware of most of these. I mean, in Denmark this year, we have seen problems with both drought, flooding, weeds, diseases, pathogens, not least in the organic production. And this is quite often what we talk most about. And we talk about soil-borne diseases as well, and we ensure that we have several years, many years, 7, 8, maybe more years between legumes in our crop rotation. So we don't talk very much about nutrient deficiencies. So I would say fertilization strategies to cereals have really been fine-tuned during the past decades. I don't think there is much more that we can gain here, but when it comes to legumes, we normally say that they are not fertilized. I mean, they are self-sufficient with nitrogen, and then in most cases we don't really do anything because we have considered legumes as a crop in a crop rotation that should fix some nitrogen and release it to the following crop. That has kind of been the main role of legumes in our crop rotations. But now we're slowly beginning to consider them as the main crop and the main crop for human consumption. So therefore, we also need to start optimizing the production much more. But unfortunately, we see unstable harvest yields, not least due to climate change. We also see the raw material quality varying quite a lot. In many years, there is a lack of economic profit for the farmers. And for human consumption, we still have a rather limited focus on legumes. So for human consumption, for plant-based diets, there are some crops that we focus more on in Denmark now compared to previously. I wouldn't say that they are new crops, but they are novel in this context. And I would say faba beans and peas are probably those that we grow the most. And then, of course, we also have chickpeas, lupine, lentils. Oat is being used for oat drinks. We still import quite a lot. We have quinoa, amaranth, buckwheat. Soybean may be entering. Right now, we are importing most of our soy and using it for feed. But with these new crops, we also have quite a few challenges because there are really large differences in their tolerance to drought, weeds, diseases, pathogens, to their requirements regarding fertilization, harvest yield, but also what we can use the crops for. So that's what we call the functionality. And also the taste, nutritional value, protein content, etc., etc. So there are lots of unknowns when starting to grow these crops and use them for plant-based diets. Nevertheless, the nutritional recommendations in Denmark actually say that each adult person in Denmark should consume 100 grams of prepared legumes per day in Denmark. So we need to grow much more. But even if our small population in Denmark is consuming 100 grams of prepared legumes every day, we wouldn't need more than a couple of 100,000 of hectares. So we definitely have room for this in our agriculture. So now I'm slowly going to zoom in on plant nutrition and the plant nutritional aspects of food quality. So I always show my students this basic equation for biomass production and plant growth and ultimately production of food. So what you need to ensure is basically that you have light carbon dioxide, water, and 14 nutrients supplied from the soil at the right time during the growth season. But as you all know, it is a bit more complicated than that because there are quite a few other key factors that control plant growth, harvest yield, and quality. And by the way, when I say nutrients, I mean the 6 macronutrients. So we have potassium, calcium, magnesium, nitrogen, phosphorus, and sulfur. And then we have 8 micronutrients. So those are the ones shown here in orange. For example, manganese and iron. So I divide this into macronutrients and micronutrients. But there are several key factors controlling plant growth, harvest yield, and quality. And we all know about the GEM matrix, so genetics, environment, and management. Genetics is plant species, for example, the cultivar. We have differences between genotypes in their ability to take up water and utilize nutrients, for example. We have differences in stress tolerance. But the environment is actually also quite important. There are differences in precipitation, in light, CO2 availability, temperature, soil type, microbiology, and this, of course, also affects the availability of plant nutrients. And then finally, the management. Well, that's soil tillage, for example, sowing, fertilization, plant protection, etc., also affecting plant nutrition. So we have a focus on, you could say, plant nutrition in each one of these boxes, and then we also study the complex interactions between these. And I'm going to get back to an example where we study nutrient deficiency during drought a bit later. You also talk about the GM matrix when you study crop quality. So quality is actually, well, it depends a lot on who you're talking to actually, because some consumers say the main quality parameter for me is price, or it's the color, or it's the taste, or whatever of the food that I'm buying. But you can actually divide it into three different pillars here. So quite often you talk about the technical quality. So how much time does it take for you to boil a pea, for example? What happens when you start processing it? Then you have the sensory pillar here, which is about taste, of course, and flavor, bitterness, etc. And then the nutritional quality. So this is a way to separate quality into three different groups, you can say. And what controls it is the environment, the genotype, but also management. And here it's from a mineral nutrition book. The author has written mineral nutrition, and I fully agree. So when we talk plant nutrition, I mean the essential plant nutrients, the six macronutrients and the 8 micronutrients. I'm going to get back to this. But when we talk human nutrition and food quality in relation to this, we divide these compounds into different categories. So you would quite often look at food quality in these different bulk components, you could say. So if you're evaluating the raw material quality for plant-based diets, you would look at the content of water, protein, fat, fiber, carbohydrate, vitamins, minerals, etc., but also potentially toxic compounds, you could say, anti-nutrients, lectins, lysine, conlysine, tannins, etc., but also microbiological and chemical toxins. So it could be mycotoxins, for example, but also heavy metals. So screening for food quality is a pretty complex task, and you need to ensure that you cover these aspects. And if you talk to a farmer about plant quality, the farmer may be more interested in how the plant appears during growth, and this is of course also important. So here I'm talking more about the performance. So we're actually evaluating quality both from, you could say, an agronomic performance perspective, but also quality in relation to the harvested product. And the plant quality is very much dependent on how the plant appears in the field. How uniform is it? How uniform is the germination, growth, maturing? Is it easy to harvest? Is it resistant to diseases, etc.? How's the nutrient neutralization? And also, I mean, when we're talking about legumes, is nitrogen fixation running at its optimal and providing high biomass production, but also a high protein content? So I just want to briefly mention one project where we are looking into the agronomic performance, but in particular the quality of the raw material. So this is a project that has been running for a couple of years. And here we have, in collaboration with plant breeders and quite a few different stakeholders from the whole value chain, we have screened 10 different commercially available varieties of oat, peas, and faba beans. So we have grown them in the field. We have evaluated their agronomic performance, and then we have harvested them and studied the raw material quality. And we have used a number of different techniques for this. In the field, we have used non-destructive methods, chlorophyll fluorescence, near-infrared spectroscopy. And then we have used more traditional methods when it comes to the lab. And then we have taken it all the way, you could say, from raw material quality to sensorics and functionality. So we are also looking at protein types, protein functionality. We are doing organoleptic tests and so on. And then finally, this project is supposed to lead to recommendations regarding the most suitable cultivars in a Danish context for plant-based diets. So you can follow this, visit the website here where we will actually present results very soon. I'm not going to focus on it today because we are not changing the plant nutrition in this study here. We are trying to grow plants optimally and then screen differences between different cultivars of oat, peas, and faba beans. But I want to focus a bit more on this here now in the remaining part of my presentation, because what we are very interested in is really what happens with both the yield, of course, but also the product quality when we change the nutrient supply, thereby also the nutrient availability to certain crops. And in most cases, we have used peas as a model plant. So in plant nutrition, it's well known that the harvest yield is a result of nutrient supply if all other growth factors are present in sufficient amounts, or if they are not limited. So nutrient supply, that could be all nutrients. You could also focus on individual nutrients. So this is quite common to see curves like this here. So you have a dramatic increase, and then at some point, it doesn't really make any sense to continue fertilizing your crop, at least not from an economic perspective, because the harvest yield is going to flatten out and then eventually decrease. So this is quite common. So if we then look at product quality, I can't think of a single quality parameter where the curve for quality follows the one for harvest yield. I mean, unfortunately, this is not really the case. This has been studied quite intensively for wheat, for example, and various vegetables, and it's quite well established that for certain quality parameters, you achieve the highest quality if you fertilize with suboptimal levels of nutrients, at least compared to what is optimal when it comes to yield. So you could say for some quality parameters, it could be iron, zinc, it could be nitrate in leafy greens. You actually get the highest quality if the plant is slightly nutrient deficient, for example, nitrogen deficient. However, for other quality parameters, this could be protein content. We also know that we have to apply quite a lot more nitrogen to a crop than what is economically optimal from a, you could say, yield perspective if you want to have a high protein content. So if you want to exceed the content of or increase the content of protein in wheat, for example, then you add a lot more nitrogen than what is required to reach optimal harvest yield. However, this is not necessarily where you get the highest protein quality, because relatively you may actually have less of the essential amino acids. And how these relations are in legumes, peas, for example, and how individual nutrients affect this is, to my best of knowledge, not very well studied. So you could say this equation here, or sorry, this graph here, and these correlations are basically what I've decided to work on for the next 20 or 30 years. I think there is plenty of stuff to do, not least because we are having 14 different essential plant nutrients. And here they are. I rarely talk about hydrogen, carbon, and oxygen because they are non-mineral, and the plant get them from air or for water. But when it comes to fertilization and soil-supplied fertilizers, we focus on the macronutrients, so nitrogen, phosphorus, potassium, magnesium, sulfur, and calcium. And then we have to focus on the micronutrients as well. You quite often have hidden disorders of micronutrient deficiencies. Manganese deficiency has been one of the major challenges in Danish agriculture. You don't see it visually until the harvest yield and quality is significantly reduced. So this is definitely one of the major challenges. But when it comes to legumes, you also have to remember that there is a beneficial. So this is not an essential nutrient because it's not essential to all higher plants, but to legumes, cobalt is also quite important when it comes to symbiotic nitrogen fixation. Nevertheless, these are the recommendations in Danish agriculture. So the fertilization recommendations to most legumes is as follows. So they are considered self-sufficient with nitrogen, but should be fertilized with phosphorus, potassium, magnesium, and sulfur. However, it's not always done. So based on this, I hypothesize that we do actually have quite a few nutrient deficiencies in our legume production out there. So if you want to understand this and try to reveal it in the field, I mean, you can actually do it for some of the nutrients if you have knowledge about the functional roles of nutrients. So this is a table inspired from a review we did back in 2020 on the molecular physiological functions of macronutrients in plants. So it's not specifically for legumes, but still it's actually valid for most plant species. So when it comes to nitrogen, I've listed the available forms here: nitrate and ammonium, the functional roles, macromolecules, proteins, chlorophyll, etc. This is quite important to know. It's also important to know that nitrogen is a phloem mobile nutrient, so it actually means that it moves around in the phloem of a plant, and this means that when you see symptoms of nitrogen deficiency in a plant, including a legume, you most often see it on the lower part of the plant. So it could look like this, for example, if you have general chlorosis, this is what we call it, at the lower part of the plant, it is quite often nitrogen deficiency due to the functional roles I've listed here. Phosphorus is also fairly easy to visually diagnose. It's important in phospholipids and nucleic acids, but also for the energy metabolism of plants. And deficiency of phosphorus leads to dark green plants and anthocyaninosis. So anthocyanins, that's kind of a sunscreen for plants, you could say. But if you have plants looking like this, very dark green, it's likely that they are phosphorus deficient. And I could continue with the other ones. Sulfur, I'm going to dive a lot more into a bit later. So it looks very much like nitrogen deficiency, but the mobility is different. So it actually means that you most often see it on the younger part of the leaves first, instead of the older parts. And I've included a photo of sulfur deficiency here. Potassium gives a different kind of symptoms. It's also phloem mobile, but you quite often have what you call marginal necrosis on the older leaves. So this is a typical symptom of potassium deficiency, whereas magnesium deficiency, which is a central atom of chlorophyll, gives you these intravenous chlorosis. I've not included calcium deficiency here. We rarely see it, at least in Danish agriculture. But I think you should be aware of the symptoms, because it's not a phloem mobile nutrient. So you actually see symptoms first on the meristem, on the youngest part of the plant, and more importantly, the roots are quite severely affected. So you have disintegration of root tissue, you have dark brown roots, and if you're working on root rot, then I think you should be aware of calcium and the calcium status of your plant, because those symptoms are quite similar. So we are studying this. We are quite interested in how the functional roles of nutrients and the nutritional status of plants is reflected in visual symptoms, but quite often you actually don't see visual symptoms. And as I'm going to show you a bit later, you may actually have significant reductions in plant performance and thereby also harvest yield without having any visual symptoms. So when it comes to legumes, we always talk about biological nitrogen fixation, or maybe it's more correct to call it symbiotic nitrogen fixation here. And most studies have focused on nitrogen in relation to this. It's quite well known that high additions of nitrogen is going to inhibit biological nitrogen fixation. But if you dive into the literature, and remember, it's all about fixing nitrogen from the atmosphere, using it for growing leaf tissue, and then the plant in return is delivering carbohydrates to the bacteria. But if you dive into the plant mineral nutrition literature, you actually find that most nutrients have a direct or indirect role in nitrogen fixation. So the nitrogen one is quite well known. Nitrogen inhibits nitrogenase. It's important for leg hemoglobin and biological nitrogen fixation. But potassium is also quite important. It's important for the delivery of carbohydrates to nodules. Calcium is important because it stabilizes cell walls. It affects nodulation and nodule structure. Magnesium is important for photosynthesis and thereby also carbon fixation. Phosphorus is important for energy supply, so it also affects nodulation. And this is also the case for sulfur, but sulfur has a more direct effect on nitrogenase activity and the leg hemoglobin content. So these are kind of, you could say, the easy nutrients to work on, because in most cases you can actually see it visually if the plant is suffering from it. At least at the end of the growth season. But it gets a bit more tricky when it comes to the micronutrients, because while we often have macronutrient contents in the percentage, we are often below 100 micrograms per gram when it comes to the micronutrients in plant tissue. Nevertheless, many of these actually are directly or indirectly involved or affecting biological nitrogen fixation. It could, for example, be via their role in photosynthesis. It could be via stabilization of cell walls, as is the case for boron, so it's important for nodule formation. Manganese, for example, is important for photosynthesis, enzyme activation, lignin biosynthesis, etc. Molybdenum is important for enzyme activation. Nitrogen assimilation, it's also a constituent of nitrogenase, which is essential for biological nitrogen fixation. And I could say the same for cobalt, which is this beneficial nutrient, as I explained previously. So it's in cobalamin coenzyme B12. So micronutrients are actually also quite important for the function and the symbiotic nitrogen fixation of legumes. And I'm very interested to discuss with you how you actually quantify nitrogen fixation. It's actually not so easy. I mean, we have tried many different things. You could of course count nodules, weigh nodules. There are also various enzyme assays and so on. But we quite often combine this with stable isotope ratio analysis. So we've been doing this for quite a few years for many different purposes. So IRMS is an abbreviation for isotope ratio mass spectrometry. And most of the plants that we grow and harvest, we run through an IRMS instrument. So we can do this because most elements of the periodic table exist as two or more stable isotopes. So if you take a plant, remove all the water, and then analyze the dry matter tissue, then you will find that more than 95% of what you have left is carbon, oxygen, and hydrogen, and then you have the essential plant nutrients in the remaining couple of percent. But we don't only analyze the total content, we also analyze the stable isotope ratios, and this is actually a pretty powerful tool for analyzing the impact of growth conditions, and not least on biological nitrogen fixation. So for nitrogen, we have two stable isotopes, 14N, that's the light isotope, and 15N, that's the heavy isotope. And you present stable isotope ratio data according to the delta notation, as I've shown here. We separate the heavy versus the light isotopes using a very strong magnet. So doing this in an isotope ratio mass spectrometer, you can separate the heavy versus the light element isotope of an element. You can then calculate the ratio between the heavy versus the light isotope, and then you can compare this to a standard. And for nitrogen, you always use air as a standard. If your delta value is above 0, so if it's positive, it means that your sample has more heavy isotope than the standard. If it's negative, it means that it has less heavy isotope than the standard. So if it's positive, we say that the sample is enriched. If it's negative, we say that it's depleted. And we use this in most of our research. So when plants conduct nitrogen fixation, they also fix nitrogen from the atmosphere. And since the standard in this delta equation is air, it means that the delta 15N value is most often close to 0 in plant tissue that has taken up most nitrogen from the atmosphere via biological nitrogen fixation. However, you should be aware that there are several processes in plants and enzymes and so on that do fractionation against either the heavy versus the light isotope. So you can actually have variations in the isotopic signature. So I'm going to show you some pitfalls a bit later regarding this. And then you may also have a very, very different isotopic signature of the fertilizers that you use, but you may actually also have a signature very close to the one you find when plants are doing biological nitrogen fixation. Nevertheless, we use this and combine it with other data. And then quite often on new IRMS instruments, you also get carbon data. And carbon data is very useful when it comes to studying the water use efficiency and stress, water, you could say drought tolerance of plants, because CO2 enters via the stomata. And when the whole machinery is running, plants are evaporating water, they are taking up CO2. Then we have Rubisco discriminating quite a lot against the heavy isotope of carbon. And this gives a unique carbon isotopic signature. But when the plant is stressed, for example, drought stressed or nitrogen deficient, then it will actually close the stomata. And it means that more of the heavy isotope is being used. So in drought stressed plants, you actually quite often have higher delta 13C samples. So we have used this in a few studies. So here you have an example. So we quite often grow plants in pot trials in our greenhouse, and then we supplement with field trials in parallel or afterwards. And this is a study here where we try to look at the complex interactions between nitrogen level and you could say water level or watering strategy. So here I've just presented data from the low nitrogen level, but then we have 3 water levels. So we have 30% field capacity. These are the plants here. We have 50% field capacity and then 90% field capacity. So you could say we have more drought stress here. And as you can see, the delta 13C values are actually also higher. So you get higher values here if the plant is more drought stressed. Interestingly, you actually also see an impact on the delta 15N values. So remember, if the values are close to 0, it indicates that the majority of the nitrogen in the plant comes from biological nitrogen fixation. And you can actually see the delta 15N value increasing when the plant becomes more drought stressed. And this is clearly showing that drought also have a significant impact on nitrogen fixation. And this was actually also what you heard about at the previous webinar. So it's quite common to quantify nitrogen fixation using stable isotopes, and we're interested in the protein content, the protein quality, but also where the nitrogen for the protein comes from. And I saw at the conference in Granada that quite a few used this equation here. So you can calculate the percentage of nitrogen derived from the atmosphere if you have a reference plant that doesn't conduct biological nitrogen fixation versus a legume plant with a certain delta 15N value. And this is based on basically what is shown here. So if you have a nitrogen fixing legume that only gets nitrogen from the atmosphere, well, then the delta 15N value is going to be close to 0. If you have a non-fixing plant or a legume that you prevent from fixing atmospheric nitrogen, then and you have nitrogen, fertilizer nitrogen in your growth media, then you are going to have a delta 15N signature very close to the one you find in the soil. If you then have a mixture, well, then you will have a mixture of these two signatures going into the delta 15N value. And I just want to briefly show you how we're working on this, because it has been a bit difficult for us to find a model system for this. And in many cases, I don't think the NDFA equation actually works. But we have been trying to work on soybean in hydroponics, because soybean we don't grow in Denmark, so we don't have contamination with rhizobium. So we have grown with inoculum and without inoculum. And as you can see here, these are the plants, the green, those are the ones with inoculum. And then you have increasing nitrogen levels here on the x-axis. You have the shoot fresh weight, and you can see that it's clearly increasing. So legumes, they do actually like inorganic fertilizers. But if we then inoculate with rhizobium, then we actually see that the soybean benefits from it. The shoot fresh weight is higher in plants that have received inoculum, but then at a certain stage it actually levels off. And here you see the number of nodules per bucket in relation to the blue bars here. And you can see, even though the plants are receiving quite a lot of nitrogen, they still produce nodules, and then it's actually not beneficial for the plant both to receive a lot of nitrogen but also conduct biological nitrogen fixation. So this is indicating that you are not completely shutting down biological nitrogen fixation by adding large amounts of nitrogen. The plant is still doing this, and there is an optimum. We're trying to find this optimum now for nitrogen addition in relation to biological nitrogen fixation, and then use this hydroponic system as a model system for studying the impact of other nutrients on BNF. So time is running, and I'm going to skip this one and then go to the final part here. So that's the Peace and Love project that you heard about, because we are trying to study the impact of different nutrients and fertilization strategies to peace. And the Peace and Love project is a project based on gene bank material. So here we are looking at 300 old accessions of peas. We are doing a screening and a characterization of this material. We are looking at the protein content. We're doing a genetic characterization. And then we're actually growing many of these accessions with different organic fertilizers and trying to study the impact on the agronomic performance, but also the quality on nutrients, proteins, and amino acids, and so on. We are looking at the drought tolerance, root growth. We are doing intercropping. And then we have a company doing organoleptic tests and developing recipes and products for plant-based diets. And then finally, we're also doing demonstration. And here you have a few photos from our field P lab from last year, where Jens, our PhD student, he grew 209 accessions in the field and tried to screen them. We have huge phenotypic variability. We have huge variability when we look at certain quality parameters. We have protein contents going all the way from 16% up to 32%. And now we are trying to study the impact of fertilization strategies to selected selected accessions from this population. And the final thing I want to present is simply just a few studies we have done sulfur. It's actually Asger who's attending here, who has done this, because we are interested in the protein content, but also the protein quality. And as you may know, legumes have an inherently low content of sulfur-containing amino acids. So we would like to see if we, by changing the fertilization strategy and adding sulfur, can increase the protein content, but also the protein quality of legumes. Sulfur has several interesting functions in plants. It regulates stomata. It's important for scavenging reactive oxygen species. It affects the amino acid profile, but also the leg hemoglobin activity and nitrogenase activity. And Asger did a study where he looked at the complex interactions between drought and nitrogen fixation in plants that were supplied with different levels of sulfur. So what is interesting about sulfur is that it takes a while before you can see visually that the plant is suffering. So pretty far into the growth period, the sulfur deficient plants, you have the low level here, middle and high, they actually looked healthy. They were just smaller. So if you had a field with just sulfur deficient plants, you would probably not know that they were deficient. But when we then compared it to plants that were supplied with high levels of sulfur, we had significant reductions in biomass production. Then we induced a drought period, and as you can see here, the plants that were subjected to drought, they were really suffering here. But however, the plants that were sulfur deficient, they had such a low biomass production that they were hardly affected by the drought. Nevertheless, I think we got some pretty interesting results that we are trying to dive further into now. Here you have the total nitrogen in dry matter. We had the main effect of sulfur supply. So low sulfur is actually also going to give you a low nitrogen content. It's also affecting the number of nodules per plants. So is a drought period, but sulfur deficiency is really severe. And then you have some interesting interactions here when it comes to the shoot dry weight, and not least the pods per plant, which I would say ultimately leads to an effect on the harvest yield. So significant reduction if you have a low sulfur supply, and then an interesting interaction here if you also remove water, then the plants were really suffering. So we are trying to study this much more. Conclusions, Nelson. So plant nutritional impacts on legume performance and quality. This was just a brief overview of some of the studies we have done, but we have a lot of ongoing work. And there is no doubt that nutrient deficiencies, they have a major impact on plants in general, but also on legumes. But we know very little when it comes to the impact on food quality. We have to say that harvest yield and food quality parameters are not always positively correlated, so it can be difficult to have both. Superior food quality may actually be obtained at fertilizer levels and nutrient supplies that are either excessive or suboptimal for harvest yield. And plant nutrition and fertilization strategies should definitely be taken into consideration when we are growing plants for plant-based food. So we don't only focus on harvest yield here, but also the food quality and the raw material quality. And then we are studying the rather complex interactions between nutrient deficiencies and drought stress in ongoing studies, both in the greenhouse but also in the field during this growth season. So always interested in collaborating on this topic, feel free to contact me. So now you know what we are working on. We are also interested in collaborating on developing analytical methods for studying legumes, if this is of interest to you. And those were the words. Sorry for talking a bit too much.
It's absolutely fine, and thank you, Kristian. It was a very nice and easy to follow presentation. Now, if
would you like me to stop sharing the screen?
Oh, what do you prefer?
Okay.
I'm not sure. Maybe if there are questions that maybe require you to go back to some slide. So if some from the audience has questions, You can also raise your hand or either write in the chat, and I'll read it to Kristian. Okay, while we wait from the audience, I have a small question for you. That is, a lot of your presentation focused on crops in Denmark, and you were very careful to keep repeating this formula. So my question is, how sensitive would be the analysis for quality and eventually the selection of new varieties oriented toward quality to the environment interaction? So how general your results would be out of the boundaries of Denmark and, for example, moving from Northern to Southern Europe?
Well, I would say the analytical methods that we are applying and testing, I mean, they are universal to.
Sure.
I mean, they can be used everywhere, and we didn't invent it. I mean, we are of course also inspired by work done globally, right? I would say when it comes to, I mean, it gets a bit more tricky when it comes to the agricultural production methods and the selection of cultivars, but also plant species. I mean, we would obviously like to be self-sufficient with soybean, for example, in Denmark, because we are importing what corresponds to, I think it's 14 or 15% of our agricultural area. So this would be great. And I know that it's being tested in Germany, in Scotland, Sweden, and so on. But in Denmark, I'm not sure it's going to work due to our climate. So we would have to take some of the crops indoor in order to study them. And I also know that crops like chickpea, for example, they only work in seasons that are particularly warm, right? But as you know, it's changing, right? And it's depressing to say it, but we may actually have a climate in Denmark that you are seeing in Southern Europe now. We may have that within a couple of decades, right? So some of the crops that are challenging now, I mean, they may actually be possible to grow in Denmark in the future. But nevertheless, no matter where you are, I mean, if you want to grow plants, you need the basic growth parameters and you need some fertilizers. So without the essential plant nutrients, you're not going to harvest anything. That's universal as well.
Okay, thank you. We have a question from Aude Tixier. I'm not sure if I pronounced it right.
Yes, that's right. Aude Tixier, thank you. Thank you so much for your talk. It was really interesting. And I have a question. So I'm a researcher at INRA of Dijon, and I work on legumes too. And my question is about the nutrient deficiencies. How do you address the differences, the effect of one deficiency on other elements? You were mentioning different elemental analysis. And so how do you address this?
Yeah, that's a great question, and this is really challenging, right? Because in you know, when you go to a field, you may have multiple nutrient deficiencies, right? So in order to study the, you know, the individual effects of nutrients, I mean, my recommendation is that you have to take it indoor, right? And we have been growing plants in hydroponics for 20 years in order to study this, because if you want to study the functional roles of one nutrient at a time and you want to know how it affects various crops, then you have to do it in a very, very controlled environment, right? And if you're doing it outdoor, even though it's of course more practically relevant, right? And you have a period of drought, for example, it affects the availability of all nutrients. So if you want to study the functionality of one nutrient at a time, I think you have to take it indoor. And even there, it can be challenging. We are doing quite a lot of studies on organic fertilizers now. Jon, who is here, is the master student. We're actually working on organic liquid fertilizers. And this is not an easy task. So you may fix a problem regarding nitrogen, and then you create another one regarding calcium, magnesium, potassium, whatever. So that's why we're using hydroponics. It's pretty far from real life, but we use that to study the nutrient deficiencies. And then if you want to analyze if you want to analyze, I would say, individual nutrients, I mean, there are actually techniques that focus on the functional roles of nutrients. Quite often it's pretty unspecific if you just analyze the total content of a nutrient in plant tissue. So for manganese and phosphorus, for example, we are looking directly into the functional roles, and we do this by chlorophyllafluorescence. So if you have techniques that addresses the functional role, for manganese, it's the role in photosystem 2 for water splitting that you address. There you can actually get a very specific response on certain nutrient deficiencies.
Okay.
Very general question to a very or answer to a very difficult question, but
No, thank you. It's enlightening.
Feel free to contact me if you're interested in discussing this.
Okay, cool.
Okay. Do we see any other questions? Something in the chat. Okay, I read from the chat. Sulfur nutrition, this is from Roger, is regularly brought up as an issue, and it's used to promote a lot of micronutrient companies. In field trials, we find it very difficult to see any kind of response to either foliar nutrition or field amendments with sulfur.
Yeah.
How are you eliciting a response? Are the trials in pot only, or do you achieve this in field too?
Yes, we try to do both actually, and we actually had 2 field trials last year on peas and different pea accessions, and we even had a fertilizer and a drought experiment where we also focused on sulfur. And it didn't work last year, unfortunately. I was hoping I was able to present this year. We had a very, very dry period, which means that the sulfur that we added, it simply just, I mean, it had to be oxidized in the soil, and it didn't happen. And then afterwards it was too wet and the plant simply just died. But you are absolutely right, and this is the main challenge of field trials. I've been doing something even more tricky during my early stage of my career. I've been working on manganese deficiency, and this is an extremely tricky element in the field. And honestly, I think I've done 1 good field trial and then probably done 5 that failed. Nevertheless, it doesn't change the fact that these nutrients are essential to the plants, and we can optimize plant growth. But if you don't see an effect in the field, it's quite often because there are other growth factors that are limiting to growth. So sulfur is a macronutrient, and it's not a micronutrient. But I know that, as you say, there are many companies that are trying to say that you should add a broad range of micronutrients, and it is quite often very difficult to prove an effect of this. Manganese is one micronutrient where we actually have proven an effect, and for some of the nutrients where the soil chemistry makes it difficult to control it, could be manganese, for example, iron as well. But there we recommend foliar fertilization instead of adding it to the soil. We are slowly moving more and more towards foliar fertilization of macronutrients as well in Denmark, because we do believe that it's more efficient and you should add less nutrients. But this still has to be proven. But field trials are definitely difficult. Nevertheless, we have to do it.
Okay. Just closing to the chat, there was an interesting remark about the fact that pollution was considered to be a source of sulfur.
Yeah, I deleted this slide this morning because, as you see, I had way too many slides. But 30 years ago, we didn't have sulfur deficiency in Denmark. But then we became a lot better at cleaning our air, and we have really significant reductions in pollution from our industry. And all of a sudden, we have sulfur deficiency in our crops, especially in rape seeds. So we have to make sure that we sulfur fertilize those.
Okay.
And there was a question regarding where I do field studies. So yeah, it doesn't say much because Denmark is a small country, but we basically have field studies all over Denmark. But we do it. Yeah.
I guess it was asking Roger, where are your fish?
No, Esker is okay. Esker says okay.
Okay, but it's okay. And did you do any essay on the field before the crops to evaluate, like a priori, if you had the situation of these nutrients?
Oh yeah, that's a hot topic as well. We wrote a book chapter on this a few years ago and basically concluded that most soil analysis out there are useless.
Okay.
So it is really, really difficult, but nevertheless, there are some analysis that we do. So we are looking at the nitrogen mineralization. We measure the total nitrogen, the ammonium and nitrates. Sometimes we look at phosphorus, so that's sodium bicarbonate, sometimes potassium, magnesium, and sulfur as well. But when it comes to the micronutrients, I would be very careful with soil analysis. Okay. We actually have 2 other projects now where we are looking more into plant tissue analysis instead of soil analysis, and then we are actually trying to do grain analysis in order to estimate whether or not the plant has been nutrient deficient, and then we use grain analysis to predict fertilization strategies or set fertilization strategies for the next for the next growth season. This is a bit unconventional, but we are giving it a try now.
Okay, thank you. Are there any other questions? Okay, it appears that we are going to close the webinar. Thank you again, Kristian, for your precious contribution, your precious contribution. And I see you all in a month or so. The webinar is going up on our YouTube channel in this afternoon, later this evening. Thank you all. Thank you all again. There was okay, there was a last-minute issue from Roger. What do you consider the best data for judging deficiencies in plant tissue?
The best data for, well, it depends on the nutrient, right? But I mean, I think it's quite difficult to do soil analysis and then predict if a nutrient deficiency will appear. It actually doesn't work for most nutrients. So I think the best thing you can do is to collect leaf material during the growth season, preferably the youngest fully emerged leaf, and then do a multi-elemental analysis. But when you do this, you need reference values in order to know if the values are high or low. So the best thing you can do is actually to select plants from a good spot in the field and a spot where they don't look healthy, and then compare.
Okay.
Yeah, but that's really the best and most reliable way to do it. Yeah.
Okay. That's very practical advice. Okay, so I'm asking again if there are any last minute question. Doesn't appear so. So once again, thank you, Kristian, and thank you everybody that attended this webinar.
Thank you so much.